Yeah, absolutely. So there's generally two different types of DNA sequencers that are out there. There are sequencers that are used to resequence a genome, generally at much lower cost, if a reference genome is already known.
Gotcha.
So let's say like we did the human genome back in 2000. It wasn't really finished and still isn't finished today. There's still some centromeres that haven't been fully sequenced yet, but 99% of the genome's known. There's a large effort trying to make sure we have maps across all the telomeres and centromeres right now, and they've managed to traverse a couple chromosomes from telomere to telomere. But once you have that reference genome, since the human population is fairly inbred, we differ maybe by 1 in 1,000 letters in the genome. What that means is you can use a much cheaper sequencer to resequence other people's genomes and then just compare it to the reference. And this is usually where Illumina comes into play. They can only sequence about 100 bases of DNA, maybe out to 300. In some extreme cases, they can do paired ends or 600 base pair reads, where they do 300 base pair reads aimed at each other. And that gives you, it does really well in the human genome. You can remap and sequence people's genomes for probably under $500 now. That doesn't include all the analysis, but just the raw cost of the sequence is getting close to that point. Now, with that said, when you're resequencing and you're mapping to a reference that is fairly representative of the entire human population, you can figure out where all the single letter changes are really easily. It's a bit more challenging to figure out where entire chapters of the genome are missing with those technologies, because when the read gets shorter than the size of the variant, it's very difficult for those reads to actually detect the variant. So as you approach like 25 to 50 base pair changes in the genome, the short read systems start to stumble and miss important genetic variation. So there's been an effort to increase those read lengths, and most of those efforts have gone in the direction of sequencing single molecules of DNA so that they get a cleaner signal. The Illumina systems right now, in order to read the DNA, they first amplify the DNA to a couple thousand copies, and then when you're sequencing that particular colony, you're actually sequencing 1,000 molecules that are all marching in step down the DNA strand. And what invariably happens as you go out in read length, each one of those reactions is probably 99.9% efficient, but over time, they start to get out of phase. The molecules get out of sync, and it gets very difficult to start reading the sequence. So to get around that synchrony problem, what the direction the technology moved was to sequence single molecules, which is much harder to do because you need to be 1,000 times more sensitive. But once you're reading single molecules, you never have to worry about the rate at which the polymerase decides to read the molecule. You just let the thing run, and you get to read the DNA as the thing is copying it in real time. So the two sequencers that are probably most known for this are Pacific Biosciences sequencers, and then an upstart sequencer that's I probably have one floating around here somewhere. It's called Oxford Nanopore, and they're much smaller footprints, but they're much noisier. These things will thread DNA through a biological protein pore, and when the DNA gets threaded through that pore, the current starts to change on you based on the sequence context that's in the pore. So the thing might zip through at 500 bases a second, and as it's doing so, it's leaving a current profile that tells you some information about the sequence. They're about 90% accurate. They're getting better. It's a very quickly improving platform. But the interesting thing about that platform is it's so small, it's portable. You can bring it to coronavirus outbreaks. You can bring it to Africa.
I see. Yeah.
And then you usually need a hell of a lot more bioinformatics in the back end to clean up the signal. Those are the read length on those are unlimited right now. They have had reads go out to 2 million bases on those things. So it's a massive breakthrough on read length.
Wow.
Somewhere that's a little bit more of a compromise between the two is a system that also sequences single molecules, but the way it reads it is using a zero mode waveguide, which is basically an array of very, very small holes. And there is a phenomenon when you're trying to image molecules, single molecules in particular, which are very small, is that the problem you have with anything that uses light or fluorescence is that fluorescence is actually a fairly large wavelength. If you look at the wavelength of blue light, for instance, it's 500 nanometers, and you're trying to measure things that are angstrom. So it's kind of like shining a floodlight on top of a speck of sand, trying to watch the speck of sand. And if you have more than one speck of sand in the floodlight, you really don't know what you're watching. And so what they had to do is build these tools that focus light into a smaller diameter than the actual wavelength of light. And that's done with the technology you can see on the front of everybody's microwave oven. A microwave oven actually has this zero mode waveguide there that prevents those much larger wavelengths from getting through. But what happens at the surface of that, never put your hand directly on the surface of that thing. It won't let you because there's a screen there. But if you were to get close enough to that, there would be a zeptoliter volume of irradiation coming through there. And that zeptoliter is enough to illuminate a single polymerase molecule copying a DNA strand. So PacBio does this really elegant device where they read 8 million of these holes at a single time. And they've managed to be able to put circular molecules into this thing. So when the polymerase grabs onto it and rolls and starts to copy it, it runs it around like a gerbil wheel over and over and over again. So you end up reading the forward strand, then the reverse strand, then the forward strand. And as you do this 30 or 40 times, you eliminate all the noise. And they now have 20,000 base pair reads that are 99.9% accurate, which is like an absolute remarkable breakthrough. And we've only had that in the last 4 months.
Wow.
So much of the work that you see in the paper we put out actually used a technology of theirs that was a little bit older, that was probably 50-fold less accurate, but it had 200,000 base pair reads. We use that to assemble a few of these cannabis genomes. And very recently, we've gone back and resequenced them with this newer hi-fi chemistry that they have to improve the references even further. But it's those two main tools that I think are being used the most in cannabis right now, is that we don't have good references, and the human genome is easy in comparison to the cannabis genome to sequence. A couple of the things that make genomes easier to sequence is that if the mother and the father genomes are fairly similar, they're easier to sequence. And that's the case with humans. In the case of cannabis, there's probably a variant every 50 letters instead of a variant every 1,000 letters. So that's much more difficult to sequence because the mother and the father genomes are like sequencing two different genomes that are blended together. And that can confuse a lot of the algorithms trying to put the sequence back together. So the long reads on these platforms, and particularly the long ones that have accuracy, are what are really shining through. They're the ones that are putting the genomes together in perfect order, near perfect order right now. And that's enabling us to start to ask, can we build a single reference for cannabis where we can resort to these cheaper tools for mapping reads? And I think the verdict's still out on that. We do a lot of Illumina sequencing. The preprint that we have up there has 40 genomes that were done with Illumina mapped back to the reads that were the references that were built with PacBio. But one thing that we discovered doing this is that while there's a single letter change every 50 bases, we're finding there's an equally high rate of entire chapters being deleted. This is what people would call structural variation. And so that preprint listed about 58 million single base pair changes in the 40 genomes that we looked at. But in one of the trios that we sequenced, which is a mother and a father and its offspring, we did that all with PacBio, so we had the visibility to see these large chapters being deleted and added. And we're seeing upwards of about an eighth of the genome that's in structural variation between just an inbred cross. That's an enormous amount of genetic material that is different just by crossing siblings.
Right.
So let alone if you step out and cross a type 1 with a type 3, it's probably going to be even more diverse than that. And it's beginning to beg the question whether we should go with the old route like we did with human, where we make a reference genome and map everything to it. It might just be the time and where the technology is mature enough where you just forget the short reads altogether and just always use something like PacBio. That's a cost question. Right now, PacBio is probably tenfold more expensive than Illumina, so a lot of people want to try and leverage the Illumina platforms as best they can. But if you really want to know what's going on, a single sequel chip from PacBio, which are usually a couple grand, you can probably get a trio sequenced on one of those now. So we tend to recommend people in breeding programs, if you don't know where your parents came from and you're not confident in them, at least sequence the parents with PacBio. And then you can do all your F1s and F2s with the cheaper tools. But if you're starting with a distant reference, like the ones in NCBI, where you don't even have access to the germplasm, and they may not even be the same type of plant. Most of the references up there are type 3 plants. You're going to have a bad time. There's so much difference between those references that they're a great starting point, let's say. But I think the direction the field's going to go is in these toward a pan genome, where if you're going to build a reference, it needs to include type 1s, type 2s, type 3s, type 4 plants, so that we have all the industrial uses of cannabis in there as a reference, so we're not missing genes.
Right, not just focusing on the drug type cultivars and.
Right.
And
or the hemp one.
Right. Yeah, exactly. Yeah. And can you describe really quickly for our listeners what are type 1, 2, 3, and 4 cannabis varieties?
So this goes back to Etienne Demeyer's work and probably Dave Watson's and a few others. They classified cannabis into 5 categories. These need to be refined, but this is just a historical classification where type 1 plants are THC dominant and tend to have a 23-to-1 ratio of THC to CBD. Type 3 plants are CBD dominant plants and have the inverse relationship. Type 2s have both genes and they make both THC and CBD. And as you get into type 4s, they don't have either gene. They don't have THC synthase or CBD synthase, and they just make the precursor cannabigerol. Type 5s are broken even further upstream and don't even make cannabigerol. And so they're cannabinoid-free plants. Now, what we've come to learn after doing all the sequencing is that was a remarkable classification system they figured out, not having ever seen the genome. They figured all this out kind of the old school way with Mendel. So they don't really know, or never published where those markers are in the genome. The preprint we put out at least attempted to do the most popular ones that we find. And the most common variations we see in these cannabis genomes that map to this nomenclature system are the type 1 plants have a broken CBD gene, so they really only have THC synthase there. And whatever CBD is coming through is probably a promiscuous synthesis from the THC synthase gene. These genes are not 100% pure. They don't make only one compound. They tend to predominantly make one compound, but leak a little bit of the others. Likewise, if you look at the CBD plants, they usually have the THC gene completely deleted. And the reason why we think the nomenclature system probably needs some more refinement is that there are some rare cases where you can have a THC gene be present in a type 3 plant, but it has a point mutation in the gene that's deactivating it.
Gotcha.
But the gene's still there. And so that can throw off some testing tools. We've had testing tools in the marketplace now for 3 or 4 years that try to just look for the popular ones, the full gene deletions, but they get tripped up when there's actually a THC gene there and there's a point mutation that's killing it because the assay is not sensitive to pick up that single base change. Likewise, if both of these genes are missing from the genome, it's usually a good sign that it's a type 4 plant. And so a lot of people have been using the sequence information to breed for CBG plants right now, because when you have both THC and CBD synthases deleted, you have less synthase genes that could be leaking THC expression. And so the other genes that we're paying close attention to in that preprint are the cannabichromine genes, because those are the next closest related genes to THC synthase. And the plant, for some reasons, made like 8 of them. It has got more of those genes than the THC and CBD synthase genes. They're not heavily expressed in a lot of the tissues we've looked at, but again, we haven't looked very deep at I think to really find cannabichromine expression, you might need to be looking at different time points in flowering and maybe in veg states to really see it turn on. We happen to see the most cannabichromine expression in male plants, and I don't know why.
Oh, interesting.
That was the only place we actually got RNA expression in that study, were in male flowers. And the Jamaican lion plants that we sequenced have a cannabichromine cassette. A lot of plants don't even have that cassette. So, but that's another gene that people like to keep their eye on. They're trying to breed a wider spread between a cannabinoid and THC that's compliant with the USDA regs. They pay very close attention to the copy number of THC synthase, CBD synthase, and CBC synthase, and that usually gives you some guidelines as to how much you might leak.
Yeah.